This sculpture is made out of the building material known as Lego. But what are living things made out of? In this video, we'll be looking at the 4 different types of materials that are used to construct living things: the carbohydrates, lipids, proteins, and the nucleic acids. These are collectively known as biomolecules. These are molecules which have been created by living things for the purposes of that living thing, whether it be repair, growth, or reproduction. It shouldn't be too surprising then that the materials that we need, the biomolecules, come to us in the form of our diet. Most of these biomolecules are listed on the back of food items. The first of these biomolecules is the carbohydrates. These are found in the sugar component of your diet. Now, the smallest possible unit is what we call a monosaccharide. This is things like glucose, fructose, or galactose, which you can kind of think like a single building brick. Now, when you add 2 monosaccharides together, you get what's called a disaccharide. An example of this would be lactose, a sugar that you get in milk. If you're like most of the world, your body doesn't produce the enzyme lactase, which will digest the sugar into its 2 components. As a result, it gets passed on to the intestines, where gut bacteria will process it and produce carbon dioxide in the process. And it doesn't come out pleasant. Anyway, that's an example of 2 sugars. And when you add even more, you get what's called a polysaccharide, which you have poly meaning many parts, or many sugars. What's interesting is that individual sugars your body will use for energy, typically glucose, but other sugars can be broken down into short-term energy as well. People who typically plan to use a lot of energy in a short time will load up on carbs for that process. In addition to energy use, carbohydrates can also be used for communication within the body. The roughly 30 trillion cells of your body are all coated in a sugar. But it's not just a simple sugar, it's a combination of many different sugars. It's a polysaccharide. The arrangement of the monosaccharides in the polysaccharides expressed on the surface say a different message. Different monosaccharides into one structure will send one message, but if you have different monosaccharides or different arrangement of monosaccharides, it will communicate a different message. And different cells will have different polysaccharides on its surface, sending different messages. There's a paper that goes into more detail where they use artificial intelligence to learn what the language of these carbohydrates is. It's a fascinating read, and I'll put it down in the description box below. Now, the next of the biomolecules is what we call lipids. These are the fats and oils of your diet. Think butter or bacon. These are also used for energy storage, but instead of being used for short-term energy storage, it's used for long-term energy storage, as it takes more effort to burn. Think, for example, when you have excess calories, it gets stored as fat in, the fatty areas of your body. This is a special tissue that holds onto the fat called adipose tissue. It stores up fat for use at a later time when you're starving. Your body will deplete all of the carbohydrates and then start to burn the longer term energy storage in the fat. But it's not just energy storage that lipids are useful for. They're also used structurally. For example, the outer boundaries of your cells are made from these lipids. These form a boundary that keeps the good stuff in and the bad stuff out. And it's also used to form structures within the cell. Think things like mitochondria or the nucleus, or endoplasmic reticulum, or the Golgi apparatus. These structures within the cell also have their boundaries formed by the lipids. It's not just the energy storage and structures that lipids can be used for, they're also used for communication. Yet another communication within the body. And it's done through what's called steroid hormones. Most hormones are actually the third class of biomolecules, the protein, but a small percentage are these steroid hormones. These are hormones which are made from cholesterol and modify it so that it sends a signal within the body. Yes, your body naturally produces steroids, but you've probably heard about those scandals in sports before where people would use steroids to enhance their body for performance purposes, whether it just be bodybuilding in general or athletic performance in the Olympics or something like that. The next biomolecule is the protein. Now the next of the biomolecules is the proteins, and it's not so much used for energy, but it can be used for energy with really desperate times. It's got a host of other functions, such as structural functions, like building the structural support for the cell. This is things like the protein laminin, actin, tubulin. Then you've also got things like mobility or transport, where you have things like kinesin. This will transport items from one end of the cell to another. And then you've got other structural mobility proteins which are used in muscles, which help with the contraction process. You've also got cell transporters. This helps, say, get the good stuff into the cell or help to remove bad stuff out of the cell, or sometimes both at once, like the sodium-potassium ion transporter. And then you've also got things like enzymes, which help make a reaction to happen much quicker. These can get really efficient, and they help chemical reactions that otherwise wouldn't happen to happen. This is really important for biological structures, where you need one particular form of a molecule to be made, whereas if it were done chemically, you'd have multiple different variants of the same molecule, which are still chemically the same, but structurally different. An example of this would be thalidomide, a drug which was used for treating morning sickness. However, one variant which was actually treating the morning sickness was good, but the alternate variant of it was actually causing birth defects in the children that were born. Structure is important. Another function for protein is neurotransmitters like adrenaline or dopamine, or hormones like insulin. This is also another communication amongst cells within the body. Insulin, for example, tells your body to take up glucose that is in the digestive tract, so that the body will use up the glucose that is available. The point I'm trying to get across here is that there's lots of very different uses for proteins. Now that said, although proteins themselves aren't necessarily used for energy, they do help with the process of extracting energy from the other molecules, whether it be lipid or carbohydrates. The smallest building block of a protein is what is called an amino acid, and there's 20 different variants of these. And they basically have the same core structure with a different side chain. Now, as you attach these together, you get what's called a polypeptide, or many peptides. The proteins, the polypeptide structure, has a primary structure, which is the sequence of the amino acids in order. The secondary structure is the common themes. You have the alpha helixes and the beta sheets, which sort of go one way and back and back. And you've also got these random coils, which are free form. And then after that, you have your tertiary structure, where your alpha helixes and your beta sheets and those three loops are arranged in some 3D arrangement. You can also sometimes get quaternary arrangement, the fourth structure, in which you have multiple different proteins forming together to form a structure. For example, hemoglobin has 2 alpha chains and 2 beta chains, or 4 different proteins, that 2 of one type, 2 of another type, but it's 4 proteins that join together to make the structure of hemoglobin. And what's cool is that proteins can be used to synthesize the other biomolecules. For instance, proteins can be used to synthesize carbohydrates from scratch in the case of plants or in the case of animals, be able to join different monosaccharides together to form a different polysaccharide, and do so in such a specific way that you can't do with chemicals mixing together in randomness. In the case of lipids, protein can be used to join the fatty acids together with the glycerol component, or even synthesize the fatty acids, and in doing so creates a lipid. In the case of nucleic acids, which we'll talk about later, protein can be used to synthesize the building block for the nucleic acid, or attach the different individual monomers into the polymer of nucleic acids. Proteins are great at building these other biomolecules, whether it's synthesizing the individual monomers or putting the monomers together to form the polymers. But proteins don't have the information within themselves to make more proteins. To do this, they need information storage within the nucleic acids. And so this brings us to the last of the biomolecules, the nucleic acids. This is your DNA and RNA. In fact, the Na in both of these structures refers to nucleic acids. The full name of DNA is deoxyribose nucleic acid, and RNA is ribose nucleic acid. The difference between these two structures consists of the backbone part, and you get it in your diet, just not in large enough quantities to be showing up on your food label. But you get it in anything that was once living, whether it be fruits and vegetables or meat. Now, nucleic acid's primary function is for the storage of information, whether it be long-term in the form of DNA or temporary in the form of RNA. The monomer for the nucleic acid, the nucleotide, can be modified so that it can store energy for use within the cell. For example, the monomers of nucleic acids, called nucleotides, you have 4 different variants, A, T, C, and G, adenine, thymine, guanine, and cytosine, can be modified so that energy can be stored in these modified variants. The most common of these is ATP, or adenine triphosphate. Typically, glucose is broken down and the energy is stored in ATP so that it stores the energy within these phosphate groups for quick use around the cell, and it's used just about everywhere. But other variants can be used as well, like guanine triphosphate or GTP, but that's a bit too nerdy. So the information storage can be stored through the use of these 4 nucleotides. Depending on the information in one strand will give you the information in another strand. Then what you can do is separate those 2 strands and construct new strands for each of those separate strands. For example, the information on one side of a DNA strand will be complemented by a new strand. So as a new strand is constructed, A will bind to T, or T will bind to A, G will bind to C, and C will bind to G. And so that's how you get information spreading through the use of DNA. Now, DNA is housed within the nucleus of a cell, and the information which is in the DNA needs to leave the nucleus so that it can make it to what are called the ribosomes. These are kind of like kitchens or manufacturing plants of the cell, as they'll be manufacturing proteins for the various functions that proteins do. Now, the information for making the protein is coded within the DNA. To get that information out, part of the DNA, which we call a gene, will be copied. And it will be copied not onto DNA but onto RNA. And this is what we call messenger RNA, as the message that this RNA contains will then leave the nucleus, enter the ribosome, and it tells the ribosome which ingredient to use and in what order. And in doing so, the ribosome manufactures the protein using the information from the messenger RNA. And the way that messenger RNA is able to convey the information to the ribosome which ingredient to use is what we call the codon list. As there's 4 different nucleotides, there's 4 different combinations for each particular site. However, there's 20 amino acids. So what happens if you get 2 nucleotides? Well, with 2, you get 16 different possible combinations. That's still not enough to produce the information required to say the 20 different ingredients that are required for proteins. So instead what we have is 3 different nucleotides. Now that said, it could all be the same AAA or TTT or GGG. You have a combination of 3 nucleotides make what we call a codon. And the combination of those 3 specifies a particular amino acid. And it does this by the use of transfer RNA. Transfer RNA is RNA that forms a particular 3D structure and also has a particular amino acid associated. So if you have the messenger RNA, say AAA, there's a transfer RNA, that's TTT, and will bring the specified amino acid into the ribosome. Then the ribosome will then move that along and read the next codon, and then grab the specified transfer RNA for that and move that in. And as it does so, it brings the amino acids on the end of those transfer RNAs together, building the chain of the polypeptide. As it continues to do this, the protein grows until it reaches its final structure size, and then folds into its complex 3D structure that is the final protein. After that protein has been manufactured, the messenger RNA will usually disintegrate or fall apart, and go back into its constituent ingredients. And the transfer RNA goes back to get replenished with an amino acid for that particular one. But it requires the protein to do that for each particular amino acid. Now, there's other different types of nucleotides. For example, the ribosome is a mixture of both protein and RNA. Other variants of RNA also include regulatory elements, nucleotides which help regulate the expression of mRNA. So in doing so, what it does is it tells how much to make, not just that it needs to be made, but you need to make lots of these things. Other regulatory elements can help just alter the instructions so that it makes a slightly different variant. Kind of think of it like a gene coding for a hamburger, but then the regulatory element tells you not to put lettuce in this hamburger, or not to put the patty in it. And in doing so, you modify the information within it so that you have a modified version of the protein. So to summarize, we've got the 4 different types of biomolecules. Your carbohydrates, which are your sugars, your lipids, which are your fats and oils, your proteins, which you typically find in things like meat, and finally you've got your nucleic acids, which are found within things that were once living, whether it be plants, animals, or fungi. I'm looking at you people who eat mushrooms. Weird. Mushrooms are weird. And so that brings us to the end of the video. If you learned something new, please give it a thumbs up. If you had trouble with any of the different parts of it, feel free to go back and rewatch that particular part. And if you're still having trouble, please leave a comment below and I'll get back to you. Until next time, God bless. Cheers.